Okay, so welcome to the Blood Lab. This is going to be exercise 16. I'll use this PowerPoint for both the Blood and the Heart Lab, but I'm going to go ahead and just this particular video will focus on the Blood Lab. So this is always a fun lab, especially right around Halloween. Who doesn't love to talk about blood right around Halloween? It's a good timing for that. So when we look at the types of blood cells that we have, and remember, blood can be separated into the packed cells and then the plasma. So when we look at those packed cells, or what we call the formed elements of blood, right? We can break up these formed elements into the red blood cells, which we formally call the erythrocytes. And again, the erythrocytes are going to be these kind of bright red, concave, biconcave discs, because if you look on both sides, they're kind of indented in the middle, right? And they're going to function to transport primarily oxygen and carbon dioxide. So you'll see that you have quite a few erythrocytes in just a millimeter cubed of blood. You can look and you'll have about 4,200,000 to 6,200,000 erythrocytes in just 1 millimeter cubed of blood. So very small volume. And so that's the erythrocytes. Then we have all of the leukocytes, which are your white blood cells. And the leukocytes are going to include your neutrophils, your lymphocytes, your monocytes, your eosinophils, and your basophils. Your neutrophils are going to be these they have this multi-lobed nucleus inside of it. So you can see a lot of times it looks like there are kind of 3 or 4 parts, 3 or 4 lobes, we should say, to the neutrophils nucleus. And so that's a distinct factor for identifying neutrophils. Also, you'll see they have the cytoplasmic granules, which will make them look grainy in a stain slide, and they'll stain light purple. These cells are going to be primarily phagocytic, and again, they are primarily protecting us right against bacterial infections, bacteria. So, and these guys will make up about 54 to 62% of your total white blood cell count. And we have our lymphocytes. Our lymphocytes are going to be the smallest white blood cell. And if you look, the nucleus is really large. It fills up most of the cell. And these guys are also phagocytic. So they, again, are going to engulf foreign bodies. And they also are going to be what produces our antibodies. So again, your lymphocytes are going to be responsible for our B cell. And so we have B cell lymphocytes and T cell lymphocytes, and we'll talk about that more when we get into lecture. So again, that's going to make up about 25 to 33% of your total kind of white blood cell volume. Then we have your monocytes, which are your largest white blood cell here that you'll see. And they have also a really large nucleus that's kind of slightly curved, oftentimes kind of like half moon shaped. And so you'll see they're quite a bit bigger than your lymphocytes. Again, these guys are going to be phagocytic as well. And they make up about 3 to 9% of your total white blood cell count. And then we have our eosinophils. And our eosinophils are going to have a bilobed nucleus. So again, they look a lot like the neutrophils, except for they usually have these just 2 large lobes to them that are connected in the center here. And they have these cytoplasmic granules. And the big thing that will be different for eosinophils versus basophils is that they're going to stain kind of orangish red, really red. And your eosinophils are important for your response to parasites. So again, for killing parasites. So eosinophils are your white blood cell that we associate with killing parasites. And they make up about 1 to 3% of your total white cell volume. They are involved with, they do have some part in the allergic reaction as well, as I have a child that has eosinophilia, and so it can cause some problems for people if you have too many of the eosinophils. But anyway, I digress into lecture, so I'll try not to do that. And then we have basophils. Basophils are, again, going to also have a bilobed nucleus and cytoplasmic granules. However, you'll notice that these will stain dark purple or blue, very different than the red eosinophils. And so, again, your basophils are going to be responsible for releasing things like heparin. So that's what's in those cytoplasmic granules, as well as histamine. So they'll be involved in that allergic response as well. And so those are your white blood cells. Again, all of these guys are your white blood cells. And then we have the platelets. And platelets are these little fragments of cells, right? They're small cytoplasmic fragments. And their function is, of course, in hemostasis or clotting. And you're going to see you can have quite a few of them because you have 100, they're quite small. They're just fragments, and you can have 130,000 to 360,000 per millimeter cubed. So anyway, just a little bit, there's this lovely chart. You have this chart in your lab. It's table 16.1. I went ahead and put it in the PowerPoint for you as well because I will post this PowerPoint. And so you'll have all of that for you. So today we are going to do blood typing, and that will be the second portion of this video. I will actually do some blood typing for you, and you'll get to see some of this. So if we had you in the lab, you would actually do the blood typing, and hopefully one day we'll be able to send out kits, but we don't have that capability just yet. So you could do your own blood typing, but it's always fun, right, to do your own blood typing and prick yourself. That's the hardest part, is pricking yourself. But if you look, when you do your blood typing, remember that you have, and I love this nice little chart here because it kind of shows you everything. So we have the ABO system for blood typing, and on your erythrocytes, your red blood cells, you can either present with the A antigen, okay, which again is that protein on the surface of the erythrocyte, or you can present with the B antigen on the surface of your erythrocyte, or you can present with both A and B antigens. Or the fourth type would be that you present with no antigens in the ABO system. So again, if you just have the A antigen, you are type A. If you just have the B antigen, you're type B. If you are type AB, you have both, or your erythrocytes express both the A and the B antigens. And if you're like me and you're type O, your erythrocytes express no antigens, neither the A or the B antigen. So that's the antigens, or the proteins that are expressed on the surface. Then there's the antibodies. So if you are type A, obviously you don't produce type A antibodies because then your own blood would clot because your antibodies would bind to the antigens. So this is always a little bit confusing for students, but try to remember this in terms of your antibodies are going to cause, and I shouldn't say the clotting, but clumping, because I really want to establish the distinction between clotting and agglutination. Clotting, again, has to do with your platelets and clotting factors. Very different than agglutination. Agglutination is where you have these antibodies that are binding to the antigens on the surface of the erythrocytes and causing clumping. Okay? And so here you'll see that if you are type A, then you are going to produce the B antibodies because you don't have the B antigen. So you'll produce the B antibodies. I think actually really quick, what I want to do is go through what antigens and antibodies each one have. And then I'm going to go back through how you actually do the blood typing, just so you don't get too confused. So again, if you're type A, okay, you have the A antigens and the B antibodies. If you're type B, you're going to have the B antigens that are expressed on the surface of your red blood cells. And therefore, you would have the A antibodies. Okay, so if you, again, you were given type A blood for some reason on accident, that would be very bad because those A antibodies would bind to the blood cells that you were given, and it would cause clumping. And again, that would be a very bad thing. You don't ever want your blood to clump. So AB, is when you are expressing both the A and the B antigen. However, you don't have any antibodies, right? Because you're expressing both those proteins. So you have no antibodies. And then if you're my case, you are type O and you have no antigens, but both the A and the B antibodies, which means that unfortunately I cannot be given type A blood or I cannot be given type B blood. So I must be given type O blood. So again, kind of interesting. So now let's talk about how we actually test for the blood typing, which is what we're going to do next, right? So when we test for the blood typing, what we do is you're going to take your plate or your, it depends on if you are given either a card or if you're given a card that already has the serum on it. But what you'll do is you'll either drop the anti-A serum, then in the next spot, you'll drop the anti-B serum, and then we'll talk about the RH factor in just a minute, which is up here. And then that's called the anti-D serum. So that way then you can see what your blood type is, right? Because we have this RH system as well. So either we're Rh positive or Rh negative, and that the Rh is, again, another antigen that can be expressed on the surface of your red blood cells. So if you are type A, okay, then you should see clumping with the anti-A serum, but not with the anti-B serum, right? Because you don't have the antigens. So the anti-A serum is basically like a bunch of anti-A antibodies on in that serum. And so if you have the A antigens, you'll see clumping, right? But not with the anti-B serum because you don't have the antigens to bind with those antibodies. And you already have the antibodies anyway. So then here, if you're type B, you should see the clumping with the anti-B serum. So again, it's like you were given, you have that serum is anti-B antibodies that are going to bind to the antigens on the surface of that erythrocyte, and it'll cause the clumping. If you're AB, then you'll see clumping in both the A and the B tray. And then if you're like me and you're type O, you won't have any clumping in either tray, other than if you are O positive, which I am. So I am positive for the RH factor, and so I do see the agglutination or the clumping in the RH spot. So, and you'll see on the test, we'll either call that the anti-D serum or the anti-RH. It's the same thing. It just depends on, which manufacturer made the test. So what's particularly interesting then is, of course, who you can donate to. So if you are type A, then you can donate to preferably type A. You can also donate to type AB. However, again, remember that you do have some of those B antibodies, so you in limited amount. Okay. If you were type B, then you can donate to an individual that's B, or again, in a limited amount to an AB individual. If you're AB, then you can only donate to AB. Okay, because you express both the antigens on the surface. So you don't want to give AB blood to someone that's type B because their A antibodies are going to bind to the A antigens. You don't want to give your AB blood to someone that's type A because then those B antibodies are going to bind to the B antigens on the surface. And you definitely don't want to give it to someone that is type O because then they have both the A and the B antigens that will bind. So, so again, if you're AB, you can donate to only AB. If you're type O, you're what's called the universal donor. And so you can donate to type A, type B, type AB, or type O. And again, this would be the ideal is if you're type A to get blood from a type A individual. You can get it from a type O individual, but remember you do have you can get a little bit of clotting because you do have some of those antibodies. But again, that's more minor than if you were to be donating, blood with an antigen that you have antibodies for. So again, here, type O can only accept blood from type O. So we can't be given the vice versa scenario. You can't be given, type A blood, because if you're given type A blood, then you're going to see mass agglutination, mass clotting. And so when you're talking about who you can donate to, type O is the universal donor. Now when you're talking about the recipients, type AB is the universal recipient, because you can accept blood from type A, type B, type AB, or type O, okay? And again, that's because both antigens are already expressed on the surface of the cell. So you don't have you're not eliciting your own antibody response because you don't produce antibodies. And so again, you have the minor amounts of antibodies that would be given in the blood, but that's more minor than you producing your own antibody response. So that's the big thing. So here, when you were talking about recipients, A can accept from A and O, again, because O is the universal donor. B can accept from B and O. AB, universal recipient, can accept from AB, and O. And O can accept from O. O only, okay? So that gives you a little bit of information on the ABO blood typing system. So then, when you actually do your own blood typing, you're going to do something very similar to this. And so what you're going to do is you're going to take your blood sample, you're going to do the anti-A serum, the anti-B serum, and the anti-D, which is the anti-RH serum. And then you're going to mix a drop of blood into each of these. And so remember, the anti-A serum is like antibodies for the A antigen. Same with the anti-B serum is like antibodies for the B antigen. And the anti-D serum is like antibodies for the RH factor. So in this individual here, you see agglutination and the anti-A agglutination in the D here. So this individual is A positive. Okay, no agglutination in the anti-B here. So they don't have the B antigen present. So here, if you look at this one, now the anti-A is clear, but here in the B and D, you see the agglutination. So this individual would be B positive. Here in this blood sample, you see you have agglutination in both the A and the B and the D slot. So you have an AB positive individual. And here, you would have, if you have no agglutination whatsoever, you are truly being called by all of the blood banks because you are O negative, which is pretty rare. So if you are O negative, then you don't produce the A antigens, the B antigens, or the RH antigens. So you produce none of the antigens. And again, you truly are the ultimate universal donor. And now I am a case where I'm O positive. And so I am negative again for the A and B antigens. However, I do produce the RH factor. And so I do have the agglutination for the anti-D there. And so even that, again, is still, because most people are Rh positive, I still get quite a few calls from the blood banks. Yeah. So, and I don't, thankfully, I don't mind donating blood. That's one of the reasons I do offer it as an extra credit opportunity, because we are a community college and it's a great community service that you can do. So, I think that's it for the blood typing system. I love this. If you are a visual person, I feel like this is just a great image. And so hopefully I wanted to share that with you. Okay, so here we are going to do the blood typing portion of the lab. And eventually, hopefully, we will have kits to send out. So I'm going to go ahead and go through the lab just as if you were to have a kit at home. So you want to set everything out first on a piece of paper towel or something, and especially on a clean surface. I have some good old hand sanitizer here. So you want to go ahead and sanitize your hands, clean your hands up beforehand. I went ahead and set out my 3 anti-serums. I've got the anti-A serum here. I have the anti-B serum here, and you'll see the anti-RH, or sometimes it'll say the anti-D serum. And so I've got all of that. I've got my blood typing card. I have an alcohol prep pad. I have my lancet. I have my Band-Aid, which I'll actually go ahead and open so that I can be ready to use that. I also am going to use 3 toothpicks so that way then I can mix the blood with the anti-serum. And lastly, I have a glove. So what you're going to do is you're going to go ahead and you're going to glove your writing hand, whatever hand you write with. And then you are going to take your other hand. For me, it'll be my left hand because I'm right-handed. And you're going to use the alcohol pad to clean your distal fourth phalanx, right? So your fourth phalanx, remember you start with the thumb, 1, 2, 3, 4. Fourth phalanx is your ring finger, and you're going to clean that with the alcohol pad. Give it a second to dry. And then here's the tricky part, right? This is where you have to stick your finger with a lancet. So you're going to pull off the top here. Here's my lancet. And the trick to this is you have to kind of hold your hand down, because intuitively your hand is going to want to move away. So I always tell my students that your hand kind of has a mind of its own, and you don't know that until you try to prick it. So what you want to do is you want to put your hand down, and you want to go ahead and put your pinky finger and your ring finger on the finger, and especially on your proximal phalanx on that ring finger. And so you can kind of hold it down, and then you're going to go ahead and you're just going to push on the side of your ring finger. Okay, you're just going to go ahead and push down. Okay, and then the tough part for me is you have to milk it. I'm a clotter, so you can see here I'm going to really get a nice drop of blood, and then you're going to go ahead and you're just going to stick that drop of blood on each section of the blood test card. And hopefully you can see this. I'm not able to see this as it's videoing because I have this on my computer. So, and then you're going to go ahead and stick that down there. So I have a nice sample of blood here on each one. And you don't have to quite do quite as much as I did. I was really trying to get a good sample to show you. So, and then you're just going to go, I usually have a paper towel or something to kind of clean off my finger. And then of course you're going to put your Band-Aid on so you don't continue to bleed everywhere. And then we're going to go ahead and add the anti-serums. So you'll see your blood will start to kind of clot a little bit on its own, but remember there's a difference between clotting and agglutination, right? So we all should have clotting factors in there. It should start to clot, but agglutination will form large clumps. So that'll be the difference. So just because I set this backwards for you guys, here is the anti-A serum. So I'm going to do a drop of the anti-A serum, just 1 drop. I'm going to do 1 drop of the anti-B serum. Just 1 drop there. And 1 drop of the anti-RH, or again, you'll see also it's called anti-D serum. So just 1 drop there. And so now what we're going to do is I'm going to take my toothpicks, and I'm going to go ahead and mix. And I have 3 toothpicks because you don't want to mix the two, right? I don't want to mix the anti-A serum with the anti-B serum because that could ruin your test results. So I'm going to go ahead and mix each one. And I happen to know my blood type, which is kind of nice ahead of time. Some of you may know your blood type if you've donated blood before. Okay, and here's the anti-D. And sometimes it actually helps if you kind of spread your blood out a little bit. So I'm just going to spread this one out a little bit. So you can see that one a little better. And remember, again, you don't want to cross-contaminate with your toothpicks, because if you cross-contaminate the serums, then that will mess up your results. So it'll take a little while. I'm going to go ahead and I will crop out the section while we're waiting here, because it does take a few minutes before you can see this. But what should happen, right, is that if you have the antigen, your anti-serum that we put, the drops that we put on this card, are going to act as like the antibodies. Okay, so I'm going to go ahead and do this on my husband, because I know that I'm type O, and I believe he's type A, but we're going to find out because I can't remember. So again, I'm just going to go ahead and alcohol swab his 4th distal phalanx, and he's going to be okay with this, I think, maybe. I'll let it dry. I have to do it, yeah, I have to do it on the side of it. Okay, so again, I'm just going to quick prick them. Oh, it's over. Look, there you go. And then you remember, you have to get you have to kind of push their hand down, and you got to really milk it. And then this kit is nice because it has these little cups that you can. So again, here in this one is the anti-A serum that was actually on the card, anti-B serum, anti-D or anti-RH. And again, these two are positive for agglutination. You can see that difference in the clumping, and even as I move the card kind of back and forth a little bit, I think you can even see those clumps. So let me just move that back and forth so you can see those large clumps. And that is exactly what you want to see. So this card would be a positive.